Okay. Okay. So actually today we will do some slides for the peripheral nervous system lab. What I did, I collected all the photos from the previous 3 lectures and put them in one section so that we will recall everything with each other. So if you recall, this is the part of the nerve, okay? This is the fascicles. This is 1, 2, 3, 4 fascicles. And as I told you, there are layers. Please pay attention to these layers. So we have the epineurium. As I told you, this is the most external layer, the epineurium. We have the endoneurium, which encloses each single nerve fiber. This is the endoneurium. And we have the perineurium. Perineurium surround each fascicle. So this fascicle is surrounded by perineurium. Each nerve fiber is surrounded by endoneurium, and the whole nerve is surrounded by epineurium. These are the myelinated axon of Schwann cells and the axons. Okay, now, so these are the axon, the green rod here, and the Schwann cells that form the myelin sheath. So in Wallerian degeneration, as you remember, we said if there is degeneration of the axon, it follows by degeneration of the myelin, but degeneration of the myelin will not degenerate the axon. So this is Wallerian degeneration. It's clear here. These are the axon, the myelin sheath. We have cut in the axon. All of the myelin sheath and the axon forward will be degenerated, while if there is defect in the Schwann cells, demyelination occur, the axon will be preserved. And the regeneration will take place after that. As I told you, the space between the internode will be different, okay? Now, this summarizes the Guillain-Barré syndrome. If you remember, it follows an acute respiratory or immunization. There will be symmetrical paralysis. There will be problem in talking, respiration, swallowing, a problem in the bladder, bowel function. The patient will become ventilator dependent. The disease is very progressive within 2 to 3 weeks, and it will result in muscle atrophy and respiratory muscle weakness also. So in regeneration of Schwann cells, this an EM, electron microscope photo, that shows something called the onion pulp, which are multi-layered structures around Schwann cells. So this is arrangement of Schwann cells which lead to this structure called onion pulps, EM photo, electron microscope photo. Now, this is schwannoma. It is encapsulated benign tumor of the nerve. Encapsulated benign tumor of the nerve. This is the capsule, the inked layer here, and this is the neoplastic spindle cells. The schwannoma is composed of hyper and hypocellular areas. The hypercellular areas, the cells are arranged packed wall to wall, while in the hypocellular areas, there will be myxoid background and scattered cells. We have hypo and hyper, Antony A and Antony B. Remember that. Antony A is the hypercellular area and Antony B is the hypocellular areas. And in Antony A, we found the Verrucay body. Verrucay body is arrangement of the nuclei, palisaded nuclei here. These are palisaded nuclei with the eosinophilic cytoplasm collection in between. So Verrucay body are special arrangement of nuclei in palisaded manner in Antony A and schwannoma. Okay? The second benign tumor of the nerve sheath is neurofibromas. We have schwannomas and neurofibromas. Both are benign tumor. Schwannomas are composed of spindle cells, neuronal spindle cells, while neurofibromas, It's composed of neuronal cells and fibroblasts. So neurofibroma. Fibro, it means fibroblast. And there is collagen in between. So this is different from this. So this is one cell type. One cell type. And here we have neuronal cells and fibroblasts. So neurofibroma arranged in between collagen material or collagenous material. So we end with schwannoma and neurofibroma, the benign nerve sheath tumor. Now the malignant counterpart of both is MPNST, malignant peripheral nerve sheath tumor. It's a malignant tumor. All all features of malignancy will be appear here. So we have hyperchromasia, hyperchromatic nuclei. Look, they are dark. And we have mitosis. This is one. This is mitosis. So high mitosis, hypercellularity, areas of necrosis in other part. There's no necrosis here. And infiltrative borders. Just to remind you, the schwannomas are encapsulated while the neurofibromas are not encapsulated, unencapsulated. They are ill-defined, okay? Now, also, this photo will summarize the features of Parkinsonism. As I told you, remember the triad, tremor, rigidity, okay? Mask face, shuffling short step gait, reduced arm swing. So these are the features, the clinical features of Parkinson's 2 posture, يعني محني, okay? And the pill and roll tremor, زي ما حكينا زي اللي بيسبح. So what is the abnormality in Parkinson disease? This is the substantia nigra. So in Parkinson, there is loss of dopaminergic neuron in substantia nigra in midbrain, okay? This is an autopsy for patient with Parkinson disease. So this is the substantia nigra. Usually it's black in color, okay? It is lost in patient with Parkinson. So this layer is this, okay? Usually it is dark brown in color, and in patient with Parkinson, where there is loss of dopaminergic neuron, there is loss of dopamine, the color will be pale. So pallor of the substantia nigra in autopsy section. Lewy bodies are inclusions that are characteristic of Parkinson. These are the Lewy body, the eosinophilic material here, surrounded by halo, pale halo. This is the halo here, okay? They are cytoplasmic. They might be single or multiple in areas of gliosis. These are the glial cells, and these are the Lewy body, which are characteristic of Parkinson disease. Now, Huntington disease, okay? This is normal, and this is the disease. So in Huntington disease, we have lost autosomal dominant loss of caudate and putamen. I will show you a picture now. Remember, these are the areas of caudate and putamen. There will be shrinkage of these areas, and the ventricles are dilated. This is normal, and this is Huntington disease. So this is the putamen, and this is the caudate. So here and here, there is atrophy of this area with dilatation of the ventricle. So this is a photo of Huntington disease. Now, in vitamin deficiency, vitamin B1 deficiency, which lead to Wernicke encephalopathy, which is characterized by foci of hemorrhage and necrosis in the mammillary bodies and around ventricles. Look to these areas of hemorrhage, hemorrhagic foci, dark hemorrhagic foci, and necrosis around the ventricle and in the mammillary body. This is an autopsy for patient with the Wernicke encephalopathy vitamin B1 deficiency. This is show features of neuronal injury in the brain. Okay, so this is the CNS neuronal injury, either hypoxic, toxic, infectious. So as I told you, there is a shrinkage of the cytoplasm. The nuclei become pycnotic, small, يعني nuclei بتصير small, pycnotic, hyperchromatic, eosinophilic cytoplasm. With eosinophilic cytoplasm, so this is a photo of brain injury, acute brain injury, shrinkage of the cytoplasm, pycnotic nuclei, and eosinophilic cytoplasm. The eosinophilic cytoplasm and the extrinsic nuclei lead to something called gemistocytic astrocyte. So the cytoplasm will expand and take a bright pink hue، يعني بصير deep eosinophilic with the stout، يعني بصير في زي تشعبات من السيتوبلازم. شوفوا هون ميزوهم. So these are called gemistocytic astrocyte that result from astrocytic injury and repair. Okay, these are repairing astrocyte, but they can be found in glioma also. Now these are Rosenthal fibers. Remember the definition of Rosenthal fibers. They are thick, elongated, brightly eosinophilic protein aggregates found in chronic gliosis, repair, or some low-grade gliomas. So Rosenthal fibers can be seen in the brain in chronic gliosis, which is a result from injury. So this is the area of repair, or in low-grade glioma. For example, pilocytic astrocytoma is a grade 1 glioma. This is a characteristic feature of pilocytic astrocytoma, but we can see these Rosenthal fibers in chronic gliosis. Neurophagia, the microglial cells, the macrophages will come and start to repair the area of injury. So collection of congregate around phagocytic injured neuron، يعني عم بيجوا عشان ياكلوا الانجرد neuron. Okay, this is neurophagia. So as I told you, microglial cells, which are phagocytes of the CNS. So microglial cells are phagocyte or macrophages of the CNS. They aggregated in areas of injury. This is called microglial nodule. They come to repair the area of neuron injury, try to phagocyte the injured neuron, repair the area with fibroblast. Okay. This is multiple sclerosis. This is an autopsy for patient with multiple sclerosis. As you remembered, it is a white matter disease. Any disease of the myelin is a white matter disease. So there is a plaques, they are discrete, slightly depressed، نازلين لجوا depressed, glassy appearance، عم بلمعوا، commonly around the ventricle, near the ventricle. So this is an autopsy for patient with multiple sclerosis. So this is the breast nucleus. This is an HNE, hematoxylin eosine section of for patient with multiple sclerosis. Look to the plaques, large plaques. This is one, and this is one. So there is sharply defined plaques microscopically. The active plaques, there is ongoing breakdown containing abundant necrophage, stuffed with myelin debris. And there is also lymphocytic cuffing, okay? And the perivascular cuff of lymphocyte. The inactive plaque بصير خلص pale, quiescent، نايم، بيكون inflammation خلص رايح disappeared، بس بيكون عندنا شوية gliosis والاريا كلها بتصير replaced by fibrosis which result to these large plaques of MS. This is the lymphocytic cuffing. This is a feature of multiple sclerosis. Lymphocytes surround the vessel in layers. So this is lymphocytic cuffing in MS. And there is macrophages in the area, okay, trying to repair the myelin loss. What is this? This is Alzheimer's disease, okay? In Alzheimer's disease, we have variable degree of cortical atrophy. So there is shrinkage of the gyri and widening of the cerebral sulci. It's mostly pronounced in the frontal, temporal, and parietal lobes. So this is Alzheimer's disease. This is an imaging, MRI imaging for a patient with Alzheimer. This is normal, and this is Alzheimer. Look to the atrophy. And there is, because of the atrophy, there is compensatory ventricular enlargement. The look will be like hydrocephalus, okay? In Alzheimer's disease, we have something called neurotic plaques. We have fibrillary tangles, amyloid plaques, and neurotic plaques. They are focal spherical collections of dilated tortuous processes of dystrophic neuritis around a central amyloid, A beta core. So this is the area. This is the neurotic plaques, okay? When there are multiple neurotic plaques, there will be diffuse plaques, okay? So these are neurotic plaques of Alzheimer's disease. So we have neurofibrillary tangles and amyloid plaque. Remember, neurofibrillary tangles are intracellular and amyloid plaques are extracellular. These are features of Alzheimer's disease. Also, neurofibrillary tangles are microscopic H&E section stalk containing bundles of filaments in neuron cytoplasm. The flame shape عم بيطلعوا زي اللهب, okay? Cortical neuron here, pyramidal cell in the hippocampus, basal ganglia or forebrain, and the rough nuclei. So this flame shape is a feature of neurofibrillary tangle in Alzheimer's disease. And thank you for listening. Any question? Thank you. Any question؟
لا دكتور الله يعطيك العافية.
الله يعافيك. تفضلي. تفضل. الله يعافيك.
دكتور نزلت السلايد على التيمز؟
ريكوردينج.
تمام اوكي دكتور.
اوكي. احمد. اوكي احمد سامعني؟ اوكي. هلا بخلي احمد ينزل.
دكتورة
سامعني؟ هلا اند ريكورد. اوكي كيف اعمل